Thank you very much for the introduction. So today I will be presenting part of my thesis results titled Effect of Corn Hybrid Maturity and Water Availability on Yield Stability. So as you may know, corn is one of the most widely grown crops in the world with around 1 billion tons in the last year. But as you can see in this figure, in the last 20 years, the harvested area of corn in the US have increased by 13% and yields did it by 31%. In addition to this, we know that corn is a crop that has a high water and nutrient demand. So the development of management strategies that can help us to optimize the use of these resources are essential to keep increasing yield. So as a little bit of background, we can see in this figure that in the x-axis we have the cumulative intercepted photosynthetic active radiation, and in the y-axis yield. And as we increase the cumulative intercepted part, we increase yields, but also we increase biomass, water consumption, and nutrient consumption. In this other figure, we can see that the same yield can be achieved with two different maturities, but with early maturities will be reduced the growing season, and we can do a better management of the nutrients and water. So overall, we need to know what are the advantages and disadvantages of early and late maturity. So early maturities can allow us to do double crop or cover crop establishment in environments that have a season limited by the weather. Also reduce drying costs, reduce water consumptions, but more important is that it has higher yield stability. As main disadvantages of early maturities, we have that require higher plant population to achieve similar interception of radiation than longer maturities, and also a lower yield potential. For late maturities, one of the advantages is that we can increase carbon sequestration to a higher amount of biomass production and can have a higher yield potential. But as disadvantage, we can have yield losses in environments that have high latitude environments that has a detrimental effect of environmental conditions in the last part of the season, and also has lower yield stability. So our goal with this study was to understand the interaction of maturity and water availability to identify which maturities can increase yields and provide more stable yields. So we proposed two hypotheses. One is that late maturities have a higher yield productivity and stability in irrigated environments. And two, that early maturities increase yield productivity and stability in water-limited environments. So we use information from corn performance tests conducted between 2005 and 2017 in 3 different states that presented a different amount of corn acreage under irrigation. We also use 17,000 hybrid location and year combinations. So as you can see in this table, we use 59 locations in Arkansas, all of them irrigated. In Kentucky, 73 locations, all rain-fed. And in Nebraska, 73 locations with a 62% of irrigation. As it is common practice in the corn performance test to include a wide range of hybrids at each environment, to make fair comparisons, we group them into early, medium, and late based on the frequency accumulated by each relative maturity. So this way we can compare middle maturities in Arkansas that range from 114 to 117 days with middle maturities in Nebraska that range from 110 to 113. So we did a regression analysis between the mean performance of a group and the environmental index. And the environmental index was the difference between the average yield in an environment and the average yield by a state. So for example, if we have an environmental index of -2 tons per hectare, this means that we have average yield in one environment that is 12 megagrams per hectare, for example, and the average yield by a state is 14 megagrams per hectare. So EI represents the productivity of a given environment. So we performed these regressions, and here you can observe an example of what we did in Arkansas, where we have the environmental index in the x-axis and yield in the y-axis with the 3 groups of maturity. So we did an analysis of covariance to test the effect of maturity on the intercept, the slope, and stability. The intercept would be the average yield for each location, for each maturity in each location. The slope will be the responsive of each hybrid to improvement in the environmental conditions. And the root mean square error was an indicator of yield stability. So these tables summarize the results of the previous regressions. Here you can see that late maturities in irrigated environments, as it was Arkansas and Nebraska, increased yield over early maturities. And it did in an order of 3% in Arkansas and 6% in Nebraska. However, in Kentucky, differences were smaller, were around 1%. When we take a look to the slope or the response to improvements in the environment, we can see that late maturities increase the response to the environment. Oops, sorry. And here we see that in Kentucky, no difference were found, indicating that the response of all maturity groups were similar. When we take a look to the root mean square error or the measure of our yield stability, we see that early maturity hybrids increase stability, so reduce the root mean square error. And this is especially important in Kentucky because we can see that early maturities didn't have any difference in yield with late maturities and provide more stable yields. So this could be an advantage for producers to increase stability to make sure they will get a profit to stabilize their income. So we also test the difference between yield in different environments. So in this case, we did the environmental index equals 0 was the average for that state, and we did the 20% lower yields and 20% higher yielding environment. So here in Arkansas, results show that an increase of late maturities over early maturities at average environment in the order of 3%, and in high yielding environments, around 5% increase. When we take a look to Kentucky, we see differences between late and early maturities only at high yielding environments and in the order of 1.5%. And in Nebraska, the difference range between 5 to 6% in the 3 different environments, lower, average, and higher. So to conclude, we found that at irrigated locations, late maturities presented higher yield advantages, but with a lower yield stability than early ones. That's supporting our hypothesis 1. That in rain-fed areas, no difference were found in yield response among maturities, so the use of early maturities could increase yield stability, and that's preferred. And also that in Kentucky, advantage of late maturities were only in the high yielding environments. So this indicates the need of quantifying yield differences in irrigated environments, for example. So for this reason, we conducted field experiments. So because we want to test the effect of maturities and plant populations on yield and water use efficiency. So right now we have, well, not right now, the last 2 years, we have these experiments in the field where we have 3 hybrids within each maturity group, 2 planting populations, and irrigated and rain-fed treatments. So results of these field experiments will be presented in my exit seminar next fall. But also I found that it's important to quantify and do an economic analysis that allow us to determine the most profitable management options that farmers can apply, because there is a seed cost, irrigation cost, and fertilizer cost that are not addressed here. So with this, I want to acknowledge the Crop Ecophysiology Lab, Eric Rommel for his help with statistics, Monses Almeron and Chad Lee for the advice. I'm open to any questions. Yes. No, so the hybrids range a lot, from 90 days to 120 days, probably in different locations. So what we did was the cumulative probability functions. So we found that the 50% of the hybrids included in one state were the most common, and those were the middle ones. The remaining 50% were split in two halves, lower than that and higher than that, and we built that early, medium, and late. So can you rephrase that for me? Well, that's what they planted usually in Nebraska. Probably we are comparing northern states with southern states. So that's, no, I can go back and show the picture. So it ranged completely. So if you see Arkansas, middle maturities were from 111 to 117. And in Nebraska were around 110. So those don't overlap. But in order to make comparisons, we wanted to group what is more representative in each state for early, medium, and late. Because if not, we could be doing some bias there.
All right, I'm going to be talking today a little bit about our agro-ecosystem mesocosm facility that we constructed in 2017, 2018 to begin addressing nutrient cycling in agricultural ecosystems. And we are specifically focusing on, phosphorus. It is a macronutrient necessary for plant growth, second only to nitrogen in terms of plant need, but it is often has to be managed like a micronutrient because of its availability in the soil. So plants uptake phosphate only from the soil solution phosphate pool. Global studies have found that that pool is often at a modal concentration of 8 micromolar of phosphorus, and they have very rarely found soil solution phosphate pools greater than 10 micromolar. So these are very low amounts, but plants require a high amount of phosphorus in order to make it to reproduction. So during the growing season, the soil solution phosphate pool is constantly being refreshed with phosphorus from either the inorganic pool or the organic pool. And they estimate that the soil solution phosphate pool can be refilled up to 500 times in a single growing season. So considering that soils can have anywhere between 30 to 80% of organic phosphorus, that's representing a large portion of phosphorus that could be used in this soil solution pool. One of the ways by which plants can begin to access the organic phosphate pool is through the exudation of exudates such as organic acids that can begin to destabilize iron and aluminum oxides that have bound phosphorus. They can also exude exudates that can begin the decomposition process of soil organic matter. So we would expect that in a low nutrient condition environment, such as low phosphorus shown on the left here, that this priming effect would be much greater than when there is a higher amount of phosphorus in the environment. So because there are so many roots left behind in agricultural systems, is the purpose of our project to quantify root turnover of a previous crop that will result in the liberation of organic phosphorus under either a low or high phosphorus condition. And we are hypothesizing that the interaction of roots between the previous and current crops will result in a greater root turnover and liberation of phosphorus. We are also hypothesizing that turnover will be greater under a lower phosphorus condition. And the way in which we have established those two phosphorus conditions is through the soil series that we selected. The Mori silt loam was collected from the Spindletop farm, and it has a naturally high phosphorus level. It has 173 milligrams of Malic 3 extractable phosphorus per kilogram due to the phosphatic limestone parent material, whereas the Sadler silt loam has a much lower phosphorus content, 12 milligrams per kilogram, and that was collected from our Princeton location. We collected both an A and B horizon from both of those soils and reassembled them in the mesocosm, so we have two horizons. After the two soil series were established, we have established 4 treatments per soil series: a fallow treatment under which no plants will be grown over the next 3 years, a corn-only treatment, a winter wheat-only treatment, and a rotational treatment in which corn is followed by a winter wheat cover. They were fertilized according to AGRN1 standards as well as limed, with the exception that no phosphorus fertilizer was added. We wanted to maintain the current phosphorus concentration of those soils. They are watered weekly, and we have adjusted their watering so that the annual rainfall in Lexington is what they receive in the mesocosm facility. And then each horizon has a rhizotron viewing tube, which is this white painted tube on the side. That allows us to insert a special camera that can scan the tube in 340 degrees and collect root images. And then we additionally have a TDR probe in each horizon that is collecting temperature, water content, and conductivity measurements every 10 minutes over the next 3 years. So we're collecting a variety of parameters from these systems, including soil chemistry data, microbial function, as well as root system information and plant productivity. But in terms of today's presentation, because we just finished our first growing season, I'll be focusing on our root system information and biomass from the first year. So during the period of corn growth, we assessed their developmental stage every few days, and this graph shows the average number of days that was required for a plant to reach a particular growing stage. In the Sadler versus Mori soils, they match very well for the first 3 developmental stages. That is because they're relying on the seed phosphorus reserves in order to grow. After the seed phosphorus reserve has been depleted and they are acquiring phosphorus from their environment is when we begin to see the divergence and the number of days required to reach each developmental stage. We do have some developmental stages missing because we had some rat tailing in the greenhouse where we had high humidity and high temperatures causing the plants to grow rapidly, and they weren't unwrapping leaves, and the next set wasn't appearing. So we do have a gap in some of that data. Now, this photo shows the difference between a Mori corn plant and a Sadler corn plant at 63 days of growth. The Mori is on the left, it's this mesocosm right here, and the Sadler will be this one right here. You can see that the Sadler corn stalk is much thinner compared to the Mori corn stalk, which is to be expected as phosphorus concentration has been correlated with stalk thickness and hardiness. And at 63 days of growth, we're already in reproductive stage in the Mori with an ear in tassel, whereas we're still in vegetative stage in the Sadler. Now, at the end of the season, when they were harvested, there were differences in the biomass, as we would expect after seeing those photos. These Mori treatments had an average biomass of 350 grams versus the Sadler's 81 grams. And in addition to that, there was also a statistical difference in the number of ears the plants produced. The Mori plants grew like gangbusters and produced an average of 10 ears per plant, whereas the Sadler plant only produced 1 ear. And then we're also coupling that with that root data. So the day in which that picture where we saw the corn stalks was also a day in which I was collecting root images. So again, the Mori is on the left and the Sadler is on the right. And you can see how thin and fine these roots are in the Sadler system compared to the Mori system. And these thicker roots right here are actually the brace roots that have already begun to form in the Mori plants. Now, because they are at different vegetative stages at the different days, we're going to rewind the Mori back a little bit and compare them at the same vegetative stage. And you can see that still the Mori roots are a much more developed system. You can still see the brace roots have already begun to form. And so I collect root scans every 2 weeks, and we have an undergrad who has been helping me manually trace every single root so we can collect information on root number, root diameter, their length, and even the angle at which they are branching. And so this graph shows the number of roots in one of the horizons. The A horizons are shown with open circles, and the B horizons are shown with the squares. And just an important note is this is only the roots that we can see in contact with that viewing tube. This is not every single root that is in that horizon. So at the time of harvest or imaging number 8, the Mori treatments already had just over 400 roots in a B horizon and just under 400 roots in the A horizon. So they had 800 roots that we could see, whereas the Sadler treatments had roughly 400 in the A horizon, but a lot less in the B horizon. And that does translate to differences in total observed root length. And again, this is only what we can see. So the Mori treatments in total for the A and B horizon exceeded just over 15 meters of roots that we could trace and quantify, whereas the Sadler system only had about 8 meters of roots. So that's a difference in what area of the soil they can explore and begin to get nutrients from. Now the corn was followed with a season of winter wheat. So on the left is a picture of winter wheat in the Mori that did not have any corn growing before it. And on the right is Mori that followed that corn crop. And you can see we have a lot more yellow tissue at the bottom in this Mori winter wheat alone versus in rotation. And the rotation looks like a thicker and fuller stand. And there was no fertilization done before the wheat was grown. It was grown like a cover with no additional fertilization. And when we look at the Sadler, we see a similar picture in which when the winter wheat is grown alone without a preceding crop, we have a lot more yellow tissue at the bottom than when it's grown in rotation, and the rotation looks a lot fuller and healthier. And when we look at differences in biomass, the rotational treatment for Mori had an average biomass of 98 grams compared to 57 grams when it was winter wheat alone. And again, the Sadler had a lower biomass as we would expect due to the lower phosphorus. They had 57 grams on average when in rotation compared to 44 grams in winter wheat alone. And we're still following those with root scans. It's a little harder to see the wheat roots because they're a much finer root system, but you can begin to see some of the roots growing in the Mori versus very fine, almost very thin roots right here in the Sadler. And I've been working on tracing these. We are again seeing differences in the number of roots. So in just the A horizon in the Mori, we're seeing 150 roots for the wheat, whereas in the Sadler A horizon, we're seeing about 30 roots. So that's quite a difference. And that does correspond with differences in root length as well. We're seeing about 4 meters of roots in contact with the viewing tube in the Mori A horizon versus about 1 meter of roots in the A horizon for the Sadler. So just some conclusions. This is our first growing year, so we have 2 more cycles of growth in the greenhouse. We're seeing a departure of similar growth and development at V3 for corn. The Sadler treatments, however, are reaching reproduction, but with smaller stalks and smaller rooting systems. And the leaf and stalk mass did differ by soil series. Now the wheat reached similar developmental milestones across all treatments. The Mori treatments were fuller than the Sadler due to the higher phosphorus concentrations. But overall, the rotational treatment, regardless of if it was Mori or Sadler, was greener and more appealing than the wheat grown without a rotation. Some next steps we're looking at is we will be assessing the elemental concentration of the tissues that we have harvested to see if there are differences in the accumulation of nutrients. We'll also be assessing changes in soil fertility, including microbial function, and we'll be doing headley fractionation to assess changes in phosphorus, as well as continuing to trace all those root pictures. As of the end of the wheat growing season, we'll have collected 2,000 photos that myself and a very brave undergrad are tracing one by one. And so just as an example of what we're watching, throughout the growing season in those roots. We have 2 movies of their root growth with stages. You'll begin to see the roots grow in. This is while we're in corn, and this is for Sadler, and that is after harvest. So you'll see those begin to fade, and very fine wheat roots will begin growing in right at the side. Now, when we compare that to a Mori treatment system, we will see a lot more roots begin to grow. And now we've killed that with corn, and some of the wheat roots are growing in at the top where you'll see the blackening and decay of the previous roots from corn. So that's what we're following. I would like to acknowledge my committee members for all their help in constructing the mesocosm facility. I would like to especially acknowledge Paige Williams, an undergraduate in our lab who has been tracing those root systems with me 4 hours every week, or 4 hours 3 times a week, so a lot of work, as well as acknowledging Walter Rodas for his help in the greenhouse facility and our people at Campbell who sit on the phone with me for many hours. And I'd like to take any questions. Dr. Sparks?
So what is the difference?
The Sadler is slightly more acidic. They both, when they were collected, they were just under pH of 5, but they were limed to the same starting pH to begin.
Actually, the next speaker is Lane Allen. Can't be here. She wasn't feeling well, so we're just gonna have a 15-minute break. So anyone that wants to get up and speak, you're welcome to. If anybody has a comedy routine, wants to do like an interpretive dance.
What's up, Keegan? How does this thing work?
You hold down the top button, and it turns into that work now. I don't remember. Yeah, it's latest. I just saw it.
Oh.
It works.
Okay. I just wanted to, see kind of how well it works. I can put it there. Get that. This is strange. How do you presentation go?
Well, this graph didn't show. Why isn't it showing on the screen?
You gotta hit the present button.
I don't think it's what it is. Oh, hey!
Now it shows.
It showed. I wonder if my videos will play now.
Your video's not playing?
No, my videos didn't play. I was pissed.
How was your highlight, man?
Yeah, they don't. I don't understand. Like, look.
Codec unavailable. Maybe the data that you dragged them in on wasn't on your flash drive or something, so it can't trace it.
Are you trying to show your movie?
Yeah, it says it can't play it.
Maybe you could just do like your interpretation of it.
This
like
is a core. I'm really bummed.
The core dance.
It It says Kodak on the
Let me
barrel.
see what it looks like. Yeah, that's the problem.
It's MP4 though. I can play it straight off my hard drive.
Yeah, go for it.
Ah. Figured it out. Yay. I only got it to play 3 hours later. Yay.
Why don't you tell us what we're looking at?
I can do that. Hold on, let me, for some reason it put on repeat. Haha. So gang, if anybody was here this morning and saw my presentation and saw everything break, I got the video played now. So this is the AP horizon of a fragile soil. Hey, thanks for popping up, Michael, just now. So this is about 5 centimeter slab. It's from a cylinder. I've cut the edges off because there's a bit of a shearing effect on the plastic tubing. So I've got just a cube here, and you can see these sort of stone fragments for the more dense structures. The purplish green is roots from just this field that had natural grasses growing in it. The purple is the more dense part of the root, and then the green is probably more of a cellulose, like live structure of roots. So because this project allows you to see the density scale, I'm able to make soil sort of see-through and see just the roots. I could turn off the rocks. I could just show just the roots for this if I wanted to, but it allows me to sort of section off what I want to exactly look at. And I've already set a transfer function for this whole program, so that way I can just turn off what I want and whatnot. But yeah, it finally worked. Ta-da! 3 hours later.
So how thick of a column can you scan?
It's more, soil is more dense than people, so I don't want to say as much as like an obese person amount of soil, but I mean,
A simple number of centimeters.
you're going, well, no, see, you have to, since this is a human scanner, we have to set it to like spine function on an obese male to get it to trick it into thinking that it's scanning a person. So you just have to set the parameters a little bit larger. You probably get a resolution loss on something like huge, but also you have to factor in that you're putting like 200 pounds of soil on this cat stand bed. So this, like that picture I showed earlier, is just like maybe 10 pounds of soil.
Yeah, but how thick of a column?
It's 1 3/4. Yeah, you can go up pretty big. Yeah.
Yeah. Yeah.
You can also turn up the machine all the way till the safety settings give a warning. But The software? Yeah. It's called Aquarius. There's a few softwares that you can go, software programs that you go through. You scan it on the machine, and then you send it offsite to a PAX server, and then you download it from that PAX server onto their Aquarius program, and then you get access to every patient that's ever been at UK. And then, so I can change the opacity, I can change the color. I pseudo-colored everything, like these roots aren't purple and green, and this stone isn't actually this round gray color. So I could turn the soil into bright blue. We could have Grateful Dead soil if you want. As a reference for the older people. So, yeah.
All right. Anybody else? Anybody know any soil jet? Does the soil have to be sort of dry to be able to see the difference between the roots and the pores, and like water filled pores? Then I just need to get a good contrast between like the roots and the water filled pore.
Originally, I spent for, and I had them dry out for 48 hours.
Be interesting to put some plants.
Yeah, Michael's like, we should just grow all sorts of different.
Get like a corn stalk.
I did find some literature that people that plant potatoes.
Yeah.
Hi, today I'll be talking about some of the results from my master's thesis research titled Long-Term Land Management, Links Between Crop Rotation and Soil Biology. The goals of land management are to enhance and maintain the components of soil health in agriculture, ultimately for crop productivity. The components of soil health are composed of the chemical, physical, and biological. In the past, there has been large emphasis placed on the chemical and physical components of soil, while the biological component has generally been overlooked until recently. One such practice, long-term land management practice that we must make are the types of crop rotations to use for what crop to plant. In this photo, we can see corn that had been planted into no-till soil, and we can still see some of the wheat residue from a previous growing season, and also corn stubble that had been from 2 years past. So we have a diversity of organic inputs into the system, some of them being slower to decompose than others. All of this has an impact on the biological community, and that is often or not fully understood. Crop rotations do, compared to corn that is in monoculture, there's a yield gap. In this figure, the years of previously cropped corn is on the X, and on the Y, we see the essentially the yield gap between corn in monoculture and corn in rotation. On average, it was about a 5% yield gap in this meta-analysis that looked at 750,000 observations. And that yield gap between monoculture corn and corn in rotation increased to about the third year, and after that, the effect leveled off. However, the yield gap persists in the ongoing years when you compare it to corn in rotation. There's evidence of some of the causes of this yield gap, and in many cases, microorganisms have been either directly or indirectly involved. And some examples are plant pathogens, allelopathy, or a decrease in nutrient cycling. So the objective of my research was to understand the role of soil biology in yield decline and continuously cropped agricultural production systems. My first hypothesis is that microbial community biomass and structure will vary over time and with rotational treatment. Second hypothesis that we have is microbial community structure and members therein will correlate with yield. To answer these questions, long-term managed agroecosystems set up by Dr. John Grove in 1989 have been continuously planted in different rotational treatments. It's completely randomized block design, and we have been sampling from May 2015 to November 2018 every month. And we have been sampling also from high nitrogen plots. This was to eliminate any potential yield bump that we might see with corn in rotation with a soybean compared to corn in monoculture. We've been building a robust data set with many different variables, and we have a continuous soybean treatment, a double crop corn, double crop soybean rotation, a continuous corn rotation, corn, wheat, soybean rotation, and a sod control. The methods that we use to answer the questions is we use phospholipid fatty acid analysis as a measure of microbial community structure and also total microbial biomass. We've measured physiochemical variables through UK regulatory services. We've gotten Malic-3 extractable metals, total percent carbon, total percent nitrogen, and pH. We've also been downloading weather data from UK Ag Weather Station, and we have gotten air temperature, bare temperature, soil depth at 0 to 5 and 5 to 10 centimeters, and rainfall for each one of those months that we've sampled in. And then we've done some statistical analyses. We've used a mixed model analysis to screen our variables, and we have selected these parameters here for our models. And we have used non-metric multidimensional scaling in order to see how microbial communities are changing throughout the growing season. So the first thing that we needed to do in order to determine how microbial communities were changing in these different treatments was to first observe, to see if there was an observable yield gap in the treatment. Figure on the left, the x-axis represents the season average yield, while the y represents the treatment yield. The two treatments in this case are corn, wheat, soybean in gray line, and continuous corn in the blue. This data is from 1989 to 2015, and we can see that the continuous corn treatment has an observable yield gap compared to the corn, wheat, soybean rotation. On average, it was about 5%. Next, what we needed to do was see if there was a yield gap in our treatments that we were specifically sampling in, and then to be able to make some conclusions down the road. And we see a similar trend. This case, the double crop soybean treatment is in the orange line. The corn, wheat, soybean is the gray line. And we see a yield gap in the continuous corn, which is the blue line. So our plots are experiencing a yield gap. Next, we wanted to answer how the microbial communities are shifting over time within these different rotational treatments. That might be able to tell us something about how they might be impacting yield in these agricultural systems. So on this slide, we have two NMDS plots from the 2015 and 2016 growing season. We're going to start with the 2015 growing season. The treatments during this year are continuous corn, which separates here, the corn, double crop soybean treatment, which communities separate, corn, wheat, soybean communities, which separate, and the sod separate primarily along axis 1, which is about 50% of the variability between these groups. The differences between the corn-soybean rotation, which is in its first year of corn, was significantly different than the continuous corn community. However, they were more similar than the corn, wheat, soybean rotation, and they were always significantly different than the sod. However, when we moved into the 2016 growing season, the corn-soybean rotation now was in its second year of corn, and the continuous corn community separated out along axis 2. However, there is no significant differences between these microbial communities because they separated out primarily along axis 2, which was only about 8.2% of the variability between the data. So as we moved into the second year of corn, the communities have shifted to become more similar to the continuous corn, while the corn, wheat, soybean, and sod treatment always remain significantly different. We also have these vectors, which can tell us a little bit about what parameters were driving the differences in these communities. They have direction and magnitude, so the longer these vectors, the more weight they have on selecting the community. And in both cases, we see that all the vectors were pointing to the sod for many of the PLFA data. And that was due to sod having a greater microbial biomass in all of these biomarkers. When we moved into the 2017 growing season, the corn-soybean rotation was now in its first year of soybean. The community during this growing season was significantly different than the continuous corn community and significantly different than the corn, wheat, soybean community. However, it fell somewhere in between the two. They separated out primarily along axis 1, which represented 65% of the variability between these groups. And the sod always remained significantly different. As we entered the 2018 growing season, the double crop corn, double crop soybean treatment now is in its second year of soybean. And it was non-distinguishable or not significantly different than the corn, wheat, soybean rotation, which was in its wheat, soybean rotation year. The polygons overlaid one another, so there was no difference between these two, while the continuous corn community remained significantly different. And the sod, which are these polygons here, remained significantly different than the agroecosystem communities. So what we've seen is this shifting of the microbial communities in the double crop corn, double crop soybean treatment, that as they enter into their soybean years, the communities start to shift away from what a community might look like in the continuous corn. But during those corn years, the community shifted to be more similar to the continuous corn. So we have addressed our first hypothesis, which showed that microbial communities are changing throughout the growing season. But what we next needed to understand was which of these biomarkers might have been best correlated with yield. So we did a yield regression analysis on the average of the biomarkers throughout the season. The two biomarkers which were best predictors of yield were the AMF fungi and the general fungi. So these are two different PLFA biomarker groups. AMF form associations with plant roots. The general fungi could have more generalist roles in the soil environment. They may be decomposing, but they may also be acting as pathogens. What we saw is that the AMF fungi had a linear relationship with crop yield during these years. So as we increase AMF fungi, we get an increase in yield. However, the fungi had a nonlinear relationship. It was linear up until about 220 picomoles per gram of soil, not concentration of fungal lipids. However, after that point, there seemed to be no benefit of having extra fungi, or it was not correlated with an increase in yield. What this might be capturing is that fungi may be saturated in the system and we might not be able to capture differences at this level, or it could be fungi after a certain concentration may be no longer beneficial for the system and actually having a negative impact on corn yield. What we then decided to do is take our physiochemical variables that we've analyzed and see which of them were strongly correlated with those two biomarkers. And for the AMF, we can see all of the environmental variables that were used in our study. pH was very strongly correlated with the AMF community. It was one of the only major biomarker groups to be significantly correlated with the malic-3 extractables, which was an interesting finding, while many of the other biomarkers weren't significantly correlated. And it was correlated with temperature and precipitation for the month that they were sampled in. Fungi, on the other hand, also were significantly correlated with pH, but they were significantly correlated with total percent carbon, while the AMF were not. So this might explain a little bit of the differences between the two biomarker groups. And so this reinforces the importance of the different organic matter that might be entering the system that could be influencing different parts of the microbial community. In this case, it seems to be a strong predictor of the fungal biomass. And it was also influenced by both the temperature and precipitation. So in summary, we show that microbial communities changed with crop rotation and throughout the growing season. We showed that a shift in microbial community in the CS treatment towards the CC might have implications on yield, while shift towards a CWS treatment might be beneficial for ecosystem functionings and at best sod. The AMF and the fungi were the best predictors of yield during the 4 years of our study. Going forward, we would like to quantify specific fungal taxa that might be in these different rotations, maybe looking at pathogens that could be influencing crop yield during those growing season months. It would be interesting to fraction the carbon pools into highly decomposable, more recalcitrant types to see if that's influencing the microbial community on a more higher resolution, because we were just looking at the total percent carbon. In long term, we would like to generate land management practices that could incorporate a time-based management for the microbial community. And I would like to thank my committee members for helping me get through writing my master's thesis and providing insight on a lot of my data. Eric Rommel has helped with lots of the statistics here in my work, and all the RISA lab members. Thank you. Yes.
So the microorganisms that you found that correlate with this.
So in previous literature, the microbial community biomarkers that were most significantly influenced by crop rotation were the fungal biomarkers. So it's in line with previous research. The reason why I think that the fungi and the AMF may be most significantly influenced is maybe because of the different rooting structures that may be associated with a rotation, which might enhance AMF. Or the fungi, on the other hand, maybe the diversity of organic matter inputs might be contributing to greater niche availability for different organisms. We're looking at kind of a fingerprinting of the community, so it'd be really interesting to see if the diversity is changing between these treatments throughout the growing season.
Hello, everyone. Today, we're going to talk about the optimizing cropping system for sustainable production of red wheat in Kentucky. Historically, in Kentucky and other states, East Mississippi River have produced soft red winter wheat. Soft red winter wheat used to produce cookies, baked, and all baked products except bread. Also, soft red winter wheat has prevailed because of high rainfall climate in Kentucky that favors high yielding and low protein content, whereas bread require a high protein. Nonetheless, bakers in our region are interested in sourcing their bread flour and for locally growing wheat. So when we talk, before I'm starting to go through with my presentation, I want to walk you through the overview and background of the research method and our results. So when we talk about the bread wheat quality, two important factors we need to consider: protein content and gluten strength. Most of the studies shows that the protein is really affected by those factors. In addition, those factors is really affected by genetic background and nitrogen fertilizer. Here, this is the model of how the fertilizers is affected the quality of wheat and yield. And the fertilizers are already marked with arrows. And actually, most of the studies shows it's not only the matter of the input of nitrogen, it's also the time of application. So to tackle the issue of improving bread quality on soft red winter wheat in Kentucky, we conduct two sets of experiments on South Farm research at University of Kentucky. The first study was to evaluate the effect of nitrogen rate and different cultivars on soft red winter wheat in Kentucky. And the other study is to evaluate the effect of nitrogen timing application on different growth stages with two cultivars on soft red winter wheat in Kentucky. For the sake of the time, I'm going to focus on the first study, which is the cultivars and nitrogen rate. So the first experiments include 5 cultivars, as I mentioned earlier: Pembroke 2014, Pembroke 2016, Purple Citron, Truman, and Soissons, which is a French baguette genotype. And we have 2 systems, organic and conventional, and 2 nitrogen rate, 60 pounds and 100 pounds. Please, let's fly over our plot on South Farm. Here, there is my plot, the organic and conventional. This is the first year preliminary data. Here in this table, I'm gonna walking through the significant numbers, while cool stories in the main effect tables here. So we have overall like a significant in the organic versus conventional for the mean yield. Also, as I mentioned earlier, the most important factors for bread wheat quality, the protein content and lactic acid, SRC, which is solvent retention capacity, or we can say like gluten strength. And we have also a really cool story about the organic system in the term of lactic acid or gluten characteristics. So, if you remember, I mentioned earlier the genetic background and nitrogen fertilizers is really affected on bread wheat quality. And we have an evidence here. You can see the Soissons, which is a French baguette type. It's really significantly different than promising, I can say promising phenotype for Pembroke 2014 and 2016. To visualize the interaction between the system and trade in the term of average mean yield, so the average mean yield in y-axis and the system in x-axis, we can tell that there is a high average yield in term of system, between conventional and organic in 6 to 8, while on 100 rate, there is no significant difference between systems. Also, within the system itself, there's not much that difference, in the average of yield, also for organic. In the interaction for the average mean yield also between cultivars and system, and also here on the y-axis, the yield, bushel per acres, and here is the cultivars. We can tell that there is an average similar trend in the average mean yield between the cultivars. And I already marked that in similar arrows just to be clear with it. While in conventional, also a similar trend, for the cultivars except Soissons. In conventional, all the cultivars along in organic systems, there is no any significant value for this interaction. Here, in the percentage average of protein, which is important parameter for wheat quality, especially for bread baking quality, we can see there is a significant difference between systems and the level of 60 organic, also between 100 between the systems. Within the system itself, there is also a significant here in conventional and in organic. Moreover, 100 rates gave a higher value for both systems. Here is the really interesting result in this slide. So, in organic system, in the parameters of lactic acid, SRC, or gluten strength, we can see in organic, we can see that the Pembroke 14, Pembroke 16, and Truman had a higher average rate in this term of parameter. While in the conventional itself, the similar trend across the cultivars except Pembroke 16, which give like a lowest value. In organic, is the same. Here, we can see like the similar trend across all the cultivars except Pembroke 14, which is weak. From my point of view, I consider it like a promising. It has gave a higher, like a value of lactic acid SRC or gluten strength for this trait. Finally, in conclusion, I would say the organic production system could be a promising way to improve bread baking quality. However, more work should be done, and this trial has already been replicated this year to validate some results. I'd like to thank my supervisor, Krista Jacobson, and all the committee members who helped me a lot to perform this study. Also, thanks to staff of Dr. Van Sanford lab, John, Sandy, and the graduate student, Lisa, and JC Carmack. Our staff in the lab, Dr. Ben Brand, Jennifer Tyler, the staff of Dr. Hildebrand, Kaisu, and the graduate student, Mohammed Sadler and Mohammed Hassan, Steve Diver, the superintendent, farm, and Dr. Joseph Strock. Thank you.
So what does the lactic acid actually do? What properties does that confer to bread?
Actually, the lactic acid or SRC, which is solvent retention capacity, is responsible for viscoelastic of the dough, which is when you spray the dough, it's cold and shake back. This is the lactic acid or gluten strength responsible for. Like you can tell by when you order like a hand-tossed pizza and cinnamon. It's gluten-free. Cinnamon is gluten-free and kind of like the hand-tossed, like really you can tell like shape and really right. Yes. That's because maybe that's a good question. That's because maybe the delivery of nitrogen and that the delivery